Today we consider the structure and function of antibody receptors. We consider the Fc receptors, which bind to the Fc portion of antibodies, and we also will consider the polymeric immunoglobulin receptor that binds to and transports secretory IgA across to the mucosal surface. Fc receptors are characterized by having two domains, D1 and D2, which are very similar to the immunoglobulin fold that we saw present in antibodies. There are many Fc receptors, and they're all characterized by these two immunoglobulin folds, receptor for IgG, another receptor for IgG. So there are many receptors for IgG. This is a receptor for IgA, and this is a receptor for IgE. We'll look briefly at one example, and this is the odd one out that has a third domain as well. So in addition to this highly conserved D1, D2, we also have this third domain present in the Fc gamma receptor 1, also called CD64. And this is constitutively expressed on various leukocytes, monocytes, macrophages, and dendritic cells. And these are all ontologically related in the sense that macrophages and dendritic cells may be derived from circulating monocytes. And monocytes, after they enter the inflamed or infected tissues, they may follow various developmental pathways towards either a macrophage or a dendritic identity. In addition to this constitutive expression, this Fc receptor, CD64, can be induced on neutrophils and eosinophils, and this follows activation by interferon gamma and granulocyte colony stimulating factor. In addition, the receptor can be downregulated in response to interleukins 4 and interleukin 13. Another receptor for IgG is the neonatal fetal receptor, FcRn. And this is essential for passive immunity, natural passive immunity. We've already looked at one type of passive immunity, and that is treatment with antibodies, monoclonal antibodies, or with serum from cured post-infectious patients. As for natural passive immunity, this occurs between the mother and the fetus. During the second trimester, the developing fetus begins to generate a degree of immunocompetence, the capacity to generate an immune response. However, this is incomplete until some weeks after birth. In the meantime, the fetus depends for its adaptive immunity upon circulating IgG, which it takes from its mother. After birth, or in addition to that, then maternal antibodies are transferred to the newborn via the mother's milk. As for at the fetal stage, maternal IgG antibodies circulating in the blood with a pH of about 7.4 has to cross the syncytial trophoblast to reach the fetal blood. And in order to do that, the neonatal receptor for IgG, is shown in green here, is required for this transport. First of all, the process of pinocytosis, P-I-N-O, cytosis, is able to capture some antibodies circulating in the blood. And then these fuse with an early endosome, which contains the fetal neonatal receptor. As the early endosome develops, we reach the acidified stage, and this is just something that's standard vesicular trafficking pathway, which is understood with cell biology. The pH drops below about 6.5, and at this pH, the receptor is able to bind to the Fc portion of IgG antibodies. And this is important for separating antibodies and collecting them on this membrane surface and separating them from other components which will get directed to the lysosome for destruction. The acidified endosome, with its neonatal receptor carrying the IgG, then fuses with the fetal side of the syncytial trophoblast, allowing then diffusion into the fetal bloodstream of the antibody. Release of IgG from the receptor is based upon a rise in pH. So at physiological pH, there's no interaction between antibody and receptor. Under lower pH conditions, then these two bind. So that enables the transport and sorting and collecting of large amounts of maternal IgG and delivering it to the fetus, and with subsequent release of IgG into the fetal blood. Now, this receptor also functions in another. The neonatal Fc receptor can also benefit physiology by increasing the half-life of IgG in serum. And the way it does that is by the stabilization effect of the receptor bound to antibody within acidified endosomes. Within acidified endosomes, IgG can survive longer without degradation than it does free in the serum. So likewise, vesicles are pinched off from the surface of either endothelial cells lining the blood vessels or monocytes that circulate within the blood. And then after we have acidification and sorting, those contents of the endosome which are not required are directed towards the lysosome, while the endosome containing now a high concentration of IgG bound to the neonatal Fc receptor in the acidified endosome can be maintained within that cell for a long period of time and returned or recycled back to the cell surface for release into the blood at a later stage. And this can be actually harmful to us when various autoimmune diseases are occurring, which are based upon high quantities of autoimmune antibodies that are directed against self. There are a number of autoimmune diseases which result in nerve degeneration as well as severe skin lesions. In those cases, blocking the neonatal Fc receptor with various therapeutic drugs can be helpful in order to prevent this prolongation of the half-life of total antibodies. So it may seem strange that there's obviously a drawback with that. It means that all IgG is going to be having a lower half-life. But when there are very severe autoimmune diseases, there are benefits of shortening the lifespan of total IgG outweigh the possible harm of reduced immunity against other infectious diseases. And a number of drugs are being tested. The first one has been FDA approved in 2021, and other drugs are in the process of seeking approval and being tested. Understanding of the Fc receptor and the nature of the interaction with various structural regions within the Fc fragment of antibodies is also being used to generate improved therapeutic antibodies. And identifying various different effector functions of interaction between different Fc receptors and different parts of the Fc region of the antibody means that we can choose some effector functions over other effector functions, and this primarily takes the form of Fc silencing, where you basically knock out the ability of Fc receptors or a specific class of Fc receptor to target a specific site. So in that way we can alter the activity of therapeutic antibodies. Now, a type of Fc receptor called the polymeric immunoglobulin receptor is necessary for the transport of secretory IgA to the intestinal mucosal surface. The epithelial cells that line the mucosa have what is called an apical side and the basolateral side. And the apical side, this is where the contents of our food undergoing digestion is being passed. And this is where we have commensal bacteria, and this is where our mucosal surface may be exposed to harmful pathogenic microbes. The secretory immunoglobulin IgA is extremely important here in order to maintain immunological control at this surface, and to direct an immune response against any harmful invasive pathogens. In order for immunoglobulin to reach here, it cannot just diffuse through the membrane, neither can it squeeze its way through between the very tight junctions that hold the epithelium together. It has to be transported, and this transport occurs via receptor-mediated endocytosis from the basolateral side and transport to the apical mucosal side. And the first step in this is the capture of IgA, which is dimeric bound joined together by a J chain, as we saw before. And this capture is mediated by this Fc receptor called the polymeric Ig receptor. After binding, then the receptor-mediated endocytosis takes place. And with transport to the apical side, cleavage of the poly-Ig receptor occurs, leaving behind the cytoplasmic tail and transmembrane part of the receptor, while a small component of that receptor remains attached, and that is the secretory component that we saw in the last lecture while surveying the structure of IgA.